All right. Yeah, so I'll kind of just be following up on James's talk. If he was describing cause, I guess I'll be describing a little bit more like of the effect of that PTM we just discussed. Pretty much every time I lead a talk, I love to use this figure I got from this beautiful mini review on mycoplasma immunoglobulin evasion strategies from Jonathan Arfi and colleagues. A lot of cool stuff you can read about on your own if you want to learn about the cool stuff natural mycoplasmas do. But this top part right here does an amazing job kind of talking about some of the really interesting parts of the surface of mycoplasma cells. They do a lot of interesting tricks that probably play a big role in immune evasion, but also could just be functions of their mycoplasma enzymes being so loose. One of my favorite ones they show here is like their tRNA translation or charging rates are about a normal bacterium like E. coli, normal in quotes, has about 1 in 3,000 mistranslation rate. Some of the mycoplasmas, I think they found up to like 1 in 200 mischarging rates. So very interesting systems. But one of these things that was outlined here is that one thing that was noted pretty early about these organisms is that I may disagree with incorrectly being trafficked to the surface, but they noticed these cytosolic proteins going to the surface, noted like housekeeping proteins, and are believed to play certain roles in pathogenesis. We'll talk about that some more later. Another interesting thing to remember is another group kind of recently proposed that mycoplasma secreted proteins are actually kind of more released into their environment. And I believe in the review, they also pretty strongly imply, or at least I interpreted it, such that the distinction between certain materials that are part of the surfaceome of these organisms and then the releaseome of these organisms are actually pretty nebulous. So it makes for very interesting stuff to look for, especially with proteins. Really briefly, as James kind of mentioned earlier, I'll just kind of really quickly run through it. Protein moonlighting is this phenomenon we see where protein can have more than one function depending on what's happening. You find it everywhere in life. If you count viruses alive, they're there too. Otherwise, we'll just kind of snarkily say plus viruses. The functions beyond their primary ones are a little bit hard to perturb. We have to with our conventional molecular biology gold standard of like knockouts versus complementation, very hard to discern these. But the times that we do see it, there's a lot of interesting things happening where we can have, different quaternary structures or localization in different areas. And they're often very ancient and conserved proteins like DNA-K, enolase, EFTU, things like that. So lastly, but for my last bit of background kind of going into this, as James kind of mentioned this earlier, just a real quick reminder. We knew going into this that surface proteins seem to be modified with these rhamnose phosphate signals, or in Synth's case, mannose phosphate. We know that they have to use a precursor of a disaccharide or larger for it. And we do know, as we just mentioned on the last talk, that membrane proteins are the ones being modified. We don't really see in the cytoplasm. So my hypothesis for my dissertation, or one of them, was that moonlighting is retained in Syn3A. And so I decided to go on a hunt for these proteins. So when we go for a moonlighting hunt, we have to be very careful. Anytime you're looking for a post-translational modification, step one is you want to enrich for surface proteins, enrich for your modified proteins. In this case, because it's we know that these are specifically surface protein modifications, you can just do a pretty simple lysis with an ultracentrifugation step. Once we've pulled down these membranes, we digest them using a in solution digest, a trypsin lyc protease cocktail. I really highly recommend this Rapidgest surfactant if you ever are generating surface mycoplasma data sets. Just seems to make our data sets much more robust. Then we perform liquid chromatography tandem mass spec on these digested peptides. When you're identifying any glycan, but especially novel ones, people want high resolution. So we use 2 Orbitrap instruments in this case. I won't bog us down with mass spec theory too much, but these are the 2 instruments we use, and they both use pretty high resolution. This Velos Pro, I believe, has about a 0.5 Dalton's mass accuracy. So that's quite accurate already, but some people even say that that's not enough. So I also use this called a Q Exactive, which I believe has a mass accuracy of 0.02 Daltons, which makes people or the skeptics among us more happy. They also use some slightly different fragmentation techniques. Again, I don't really want to take up too much time talking about it, but basically they're two interesting kind of almost, I guess, complementary fragmentation techniques, and it helps us generate some more confident data set or confidences in our identifications when we're running samples on these two different instruments. So once we have these mass spectra, then we use proteomics software to analyze them. We like to use this software. It's quite expensive, but the really cool thing about it is for each mass spectra that's putatively identified to be a peptide against the data sets that we search, this software generates these things called decoys, which are basically just like, negative controls for each putative peptide identification. So it makes or will basically give us a statistical confidence that the proposed peptide sequence is truly that sequence in our data set versus just like, some other just random set of isobaric material. So basically, if our negative 10 log p score is above 20, that's about a p-value of 0.5, or sorry, 0.05 p-value that we always look for, like standard t-test kind of stuff. Obviously, a lot of our data actually comes up, that I'll be showing kind of here, we'll have p-values or negative 10 log p-values above 100. So I am quite confident that these are truly what we say that they are. So one kind of exemplary example of this would be for my synth data sets is this DNA gyrase subunit beta peptide we found right here. And just to kind of give you an overview of like our current screen through these, what we look for, of course, as I just mentioned, is getting good stats. I believe that says 92, negative 10 log of 92. So that's already amazing. We try to make sure that we get clear evidence of the modified residue being fragmented. Sometimes, or a lot of times, what will happen is the fragmentation will come along with some nearby amino acids. And usually I could say if a serine and an alanine just fragmented together. I don't think it's a particularly hot take to say it's probably the serine being modified here, but ideally I do want to get like a clear one like we get here. As James also mentioned, normally you want these like really like thin and Gaussian distributed precursor peaks when you're running through the HPLC. In this case, we get these really like loose wavy ones that certainly imply that shearing phenomenon he was talking about earlier in the HPLC. Lastly, I'm reticent to use the word consensus sequence, but I will say that there are usually nearby lysine, isoleucine, or lysine out or residues in the nearby region, even if they're not directly nearby like this one. We usually find them within like, I don't know, 5 to 10 amino acids. And I'm sure there's probably some structural stuff happening there too. As James said, we'd always love to talk to some proper structural biologists. I like to dabble, but it's hard enough to manage all the proteomics. So when we look through these data sets, as we mentioned, we kind of see like three, I guess I kind of categorize them as three potential groups. We have the known moonlighting proteins like enolase. We have things that canonically should be on or like annotated on the surface. Think anything that annotates as like a lipoprotein, these ATPases and things like that. We also believe we have some novel ones. I still want to do some more analysis before I put my full name to them. Certainly, this is not proportional. The novel ones are going to be a much smaller subset of this, but I definitely think that there are some on there that I haven't found any evidence in the literature of. So, like I said, hopefully we'll have this published pretty soon. Some examples of like some top hits I have here. I do caveat I am still running through these analyses. I was going to have more done. I got COVID last week, so but I'm about 90% sure these ones or each one of these has these modifications that we've identified on them at a minimum. As James mentioned, the glycan that's potentially being modified on these surface proteins are either a dimannose or a monomannose. Our dimannose hits, this is kind of some of the ones we show. I guess in the future when this is posted, you can pause and take a look. I obviously don't want to just run through each one of these individually, but certainly you can see proteins that are expected to be on the surface, some like cell division proteins and stuff like that. Ribonuclease Ys, I believe we mentioned earlier, actually, in the previous talk. And then also proteins that are known to moonlight, lactates, dehydrogenase, etc. We also get plenty more mono-manose hits and similar phenomenon. So it increases my confidence. Also, to point out something James had kind of mentioned earlier, we get like multiple EFTU hits. These are some of my top statistically scored ones at that. So that makes us pretty interested to see. I think in the future, I'd really like to kind of focus in on some of these really abundant surface proteins and try to, I guess, discern out how many modifications happen on one particular protein, because this is obviously like a cocktail of various surface peptides at this point. So to conclude, we detected moonlighting proteins in these ultracentrifuge membrane materials. We see the proteins we'd expect, including some known moonlighting proteins. They glycosylated with the sugar phosphate. I would conservatively say we have at least 50 proteins in our data set right now that are known moonlighters. Like James says, probably at least 30 are published, are complete publication quality with all of our metrics. And if I were a little looser, I could probably even say over 100. But stick to me on that analysis. I'll get back to you. Currently, we're kind of finishing up some data collection for publication. One thing I'd still like to do is a phospholipase de-shearing assay, just to account, for anyone that kind of doubts our shearing hypothesis. Just a nice way to keep the skeptics quiet. How conserved, or it's interesting to think about how conserved this PTM is across bacteria. Like James had mentioned, we've seen it in, I have some pretty convincing data that's in at least Bacillus cereus and some other gram positives. Perhaps it goes up to gram negatives, but it's hard to generate surface proteomics data sets for gram negatives. And it'd be interesting to think more about the modification, things that could affect the structure work. So definitely hope we get to talk with Pelletier and colleagues about this. And then functional work, your guess is as good as mine right now, but it'll be really interesting to go about. So I'd like to thank my lab collaborators and committee members for all their help throughout this, and the UAB Department of Pediatrics for funding this research. With that, happy to take any questions. Thank you very much.
Thank you, John. I don't see any hands up yet. Kim.
Yeah, I got a question for John or James. Do you guys yet know where the glycosyltransferases are located that actually do the covalent linkages?
I would really love
inside
to.
Or outside?
Yeah. We don't know actually even what genes it is. We've done bioinformatics searches for more canonical glycosyltransferases, and there's not very many. We suspect it's a novel one. One of the main reasons we think so is, or one of our main reasons that we suspect that is because it uses a disaccharide or larger as the precursor glycan. Other non-Laloir glycosyltransferases can use that, but we suspect that there just might be a different mechanism for why it needs that disaccharide or larger.
Okay.
So it's a good question.
Yeah, hi. You mentioned EFTU, and that is one of the most abundant proteins in every cell because of its duty in, translation. So what additional functions do you think it has? I know that in Archaea, it's been identified that oftentimes these guys are, they're sort of like satellites that are bringing in several charged tRNAs at once. But what are your thoughts about the EFTU?
Yeah, that's a great question. So the current kind of dogma, or I guess model of the field, is that these EFTUs on the surface help to adhere to the extracellular membrane for these pathogenic bacteria. It says it's in Synth, of course, and like I actually didn't even know about these Archaea ones. I'll have to read about that later. Synth being a non-pathogenic organism to me implies then that it's doing a more fundamental process for survival on the surface. What that is, great question. I've had many random thoughts at 2 a.m. about it, but I don't really. Your guess is as good as mine.
Okay, so it would be good, just like James demonstrated, how many of them are doing that function? Because boy, you need a lot of them. It has to take every charged tRNA to the ribosome. So in our model, we assume we have enough, but our model is made for the ideal functioning cell. So we're trying to write it that it's modular, that one could investigate some of these other things in it. But this is very interesting. Thank you.
Ingguang, can we
So
get
I
your
have
question
a
in before we
question
move on?
Sorry.
Hi, John. John, just a quick question. Are you able to capture transmembrane proteins with this protocol?
Yes, I believe so, but because of how the digestion happens, it does bias it towards the what's a hydrophilic domains of it. So you'll primarily probably bias yourself towards the not the transmembrane domains, but the actual surface exposed parts.